So we were talking here about things that are transported from one side of the cell to the other side of the cell. Now, it's easy to get lost on the details of this whole mechanism. So as I'm going along today, as I'm going to explain the things, we are also going to step back a little bit and look at the purpose for all this sorting and direction of stuff. Okay, so here is starting a new class. Let's make sure you get your points. Okay, everything is cool. So we had talked before how you get to export messenger RNA. Right? And this is the part that instead of just describing to you what you need to know about the mechanism, this is the part when we can actually get to have a little fun and incorporate some disease states within all this process, okay, within this whole process of sorting, transport, and etc. Okay, so here, when you look at how you're getting your mRNA out of the nucleus, remember that your mRNA, your pre-mRNA has to be processed. You have to cut out those intruders, right, the introns. You have to put in a cap at the 5' end, and that's the first thing that happens, right? When does it happen? Beginning of translation, right? And then you enzymatically put in the tail, and after you have that mRNA totally processed, then the mRNA is going to be exported via this mRNA exporter pathway. And there is only one exporter pathway for mRNA, but mainly we need those transportins, right, that are depending on GTP in order to bind to cargo and move to the cytoplasm, right? So you do have this mRNA exporter complex. So here we are looking at some proteins that are involved in that exporter pathway. Now, as I mentioned, there's not only one type of pathway, there are several types of pathways that you can use to export mRNA from the nucleus. I'll give you a couple of examples. One of them is called RAY1. This is the major type of export pathway. And there are other pathways that include C1, and this is a minor pathway, but is very important for export of proteins. And you see why in a minute I'm mentioning that, okay? So you have several exporter complexes that are going to serve as mediators between the cargo, in this case the mRNA, between the cargo and the nucleoporins that are making up the nuclear pore complex. And again, when RNA gets exported, it dives in with a cap, so it's wearing a little swimming cap and dives through this tunnel to go through the cytoplasm. If the mRNA is not properly processed, then it's not going to go through nuclear export. It's going to be degraded inside of the nucleus. Okay. What happens if the messenger RNA gets stuck in the nucleus? You never get the protein because you need to be in the cytoplasm to translate that protein. Okay, so problems with the export, that means your cell is not going to be able to make protein. Okay. So here, looking at this huge complex that is the nuclear pore complex, and also nucleoporins that are involved in transport, right? If anything goes wrong with the complex, you can develop diseases. So here are some examples of diseases that can develop in case you have problem with some nucleoporins. And we see here, cancers are associated with nucleoporin defects. And then you have aging. That means getting old. Some of the associated nucleoporins, if they are mutated, you get old faster. Okay. And issues with mitosis and meiosis. Now this is really cool. We could have like a whole class, I mean a whole semester explaining what happens in mitosis and what happens to the nucleopore complex. Because just picture in your mind, everybody studied mitosis before? Yes, yes. So what happened during mitosis? One cell divides into two, right? First you have DNA synthesis, and then you have breakdown of the nuclear envelope. You know what I'm talking about? Yeah. Yeah. And those nuclear pore complexes, where are they? Embedded in the nuclear envelope. So when you get rid of the nuclear envelope during mitosis, before metaphase, where all your chromosomes are lined up in the middle, right? When the nucleus disappears, where do all these proteins go? They don't just disappear, right? They don't just like sit on the side and wait for all the positive process to happen and then they'll decide to form the nuclear pore again. Actually, those nucleoporins, they assume different functions that are very important to conduce the proper segregation of chromosomes and the proper cell division. Okay, so those nucleoporins are really, really, really fascinating. Anyway, they influence a lot of processes and even like immune system type of processes and diseases. Those are associated with NUCs. Okay. So here I have this little picture showing you the nuclear pore complex. Okay, remember the little basket goes towards the nucleus, right? And the little fibers go towards the cytoplasm. And here I have this picture that shows that viruses actually figured out that the nucleus is a very important site for control of gene expression of the cell, right? Because viruses are very smart. So the viruses learned that if they block nuclear transport, meaning if they block the pore, they take over the cell function. Okay? And viruses are very good at doing that, taking over the cell function. So here I put some examples of viruses that will block either messenger RNA export or protein export, or a combination of both, in order to take over the cell function. So here you see, in this case, I'm putting here a main pathway for messenger RNA export. So here's your messenger RNA of the cell, right? And messenger RNA goes through this protein array 1, and they get together with other stuff. The nucleoporins go through this pathway and gets exported. Now look at this. There are some viruses like poliovirus. Everybody heard of polio? Yeah, yeah. How do we avoid polio? You get vaccinated, right? So poliovirus and rhinovirus. Do you know what rhinovirus, what kind of disease it causes? Common cold, yeah, yeah. How about influenza virus? The flu, right? Common cold is piece of cake, the flu, it's not, right? So those, they all block messenger RNA export. What does that mean? It means that when the virus gets there, it will interact with certain nucleoporins and it will prevent the cell from exporting its RNA. So the cell now cannot make proteins, even antiviral proteins. It cannot make antiviral proteins to fight off the virus. So the cell that originally got blocked by these viruses, the cell will die, okay? Now, besides nuclear export of messenger RNA, you have some viruses that block this main export pathway, which I mentioned before is Crom1. This one is mainly for export of proteins. Okay, for export of proteins. So you have viruses like measles and dengue virus and even HIV that will block this pathway and really mess up protein distribution, protein targeting, and some mRNA, some RNA type of export. Some messenger RNA also goes through that route. Okay. So viruses are really, really good at blocking this type of transport and taking over cell function. Okay, and you see other viruses here, for example, Ebola virus and other viruses. They're not very dramatic, right? But you see here, Ebola, HIV, polio are targeting several paths that block this type of nuclear transport. All right. So here, a bit of a step back when you're saying what does it mean when you're transporting things around, and especially what does it mean when you're transporting things through membranes, right? So we know that molecules usually they're not going to travel by themselves, right? So when you're looking to transport through rough endoplasmic reticulum or Golgi or lysosome or peroxisomes, those are all part of your endomembrane system. Okay, and the other ones that we mentioned here, those are okay, nuclear membranes. Yeah, nuclear membranes actually goes to the membrane system. But when you look at mitochondrial or chloroplasts, those are not part of this communication network of endomembrane system. Okay. All right, so here's a little summary telling you what these organelles do, just for a refresher. Okay? So let's get to the parts that are really, really important to know. Okay. So here, when you're talking about transport, right, of things that are proteins here, or even things that get into the nucleus, we see mainly three types of mechanisms that we have already briefly looked at. We have seen things that go through pores, like through the nuclear pore, right? So those are in need of adapter molecules that bind directly to the protein and allow the translocation. And then we see other types, these ones, the types that we covered before, and transport through mitochondria, right? Mitochondria and chloroplast. So we see that these types of transports, what happens is that the protein at the cytoplasm, it has to be stretched out and then squeezed through those transporters, like those translocators, and then folded up again when it reaches a destination. In contrast to importing to the nucleus, in which the protein goes the way it is folded naturally. Okay, so the protein is not getting stretched out when it is imported into the nucleus. It is just interacting with transporter molecules, but is not denatured. It keeps its original endogenous shape. Okay, so here, when the second type of transport, when you're going through the membranes of mitochondria or chloroplast, you remember you stretch out the protein, use those chaperones, and those have to use energy, right, to stretch out the protein. Then when you push the protein through those transporters, the chaperone stays out, but in order to dissociate the chaperone from your stretched out protein, you also have to use energy. So it takes energy to stretch out the protein, it takes energy to pry off those chaperones, HSP70, right, out of your stretched out protein. And then you have more chaperones that do folding inside. So this was the second way of transport. And now we are focusing on transport through vesicles. And this is the part where we're really focusing in now, this class, okay, transport through the vesicles, in which things are going to get packaged within the membrane bilayer, and below packaging units, they're going to be shipped to different places. So when we're talking about vesicular transport, you have two main forms of transport if you are involved in the plasma membrane. Either things go inside of the cell from the outside, and that is your endocyte. Things go inside from the outside, so this is your endocytosis. Or you can dump things to the outside, so this is secretion or exocytosis. Right? And start getting into the details. So this is from your book. showing that you are going to make special little vesicles that will have a way of knowing where they have to go. So this way of knowing has to do with proteins that are going across this membrane, okay? And they are going to sense receptors on the target compartment. Okay. This is figure 13.2. All right. So in this picture here, this is the overall map for secretory and endocytic pathways. So this is telling you about exocytosis and endocytosis when you talk about vesicles. Okay, so what is going on here? First thing, when you have a protein that is destined to either be at the membrane or to be secreted, remember, or to go to lysosome, that protein is going to be made in the rough endoplasmic reticulum. So you're going to have the signal peptide that's going to bring the protein to the ER, right? Then after that, many things can happen to the protein. It can go into the Golgi and then be processed in the Golgi, like glycosylation, phosphorylation, etc., and then get packaged and then go straight from Golgi to exocytosis. Or it can be packaged and be sent to little vesicles and then go to exocytosis, right? So it can either go straight from Golgi or go through vesicles and be exocytosed. Okay. Or it can go to endosomes and follow different routes. They're going to go back to places inside of the cell. Okay, so if you look at this map here, you'll see the basic movements that those proteins can go through. So here, a bit of a recap on what is meant by the way or by the direction of those movements of vesicles, right? So if you have vesicles that are going from ER to Golgi to exocytosis or to specific endosomes, you call this movement here anterograde, right? If you have things that are going back to the ER, you call that retrograde. So everybody got that? Just review. All right. And let me see. All right, so here, basically, we are going to start talking about the mechanism that these vesicles are being moved around, okay? So they are going to need specific markers. They're going to tell them where to go. Okay, so let's get to the nitty-gritty stuff. Okay. So here we go. Basically, you have three ways of transporting those vesicles. The first one, that has to do with anterograde transport. The first one uses specific types of coating proteins that are called COP2. So those are going from the ER to Golgi, and then later on, these cells are going to go and follow whatever destination they have to go. Okay, so the first way of transporting things is by COP2. Okay, so this is your anterograde process. The second way here is by COP1. So that one goes back to the ER. Okay, so this is your retrograde process. And the third one is by Clatrin, right? It's no COP3. Okay, I wish it was COP3, but you know, Clatrin. That's it. So Clatrin is not involved in this area over here. Clatrin is going to be involved in this section here that is basically going between the Golgi and lysosomes, or between the plasma membrane and lysosomes. Okay, so you have COP1, COP2, and Clatrin. Okay. Now here comes the fun part. There, like the analogy. How do you remember which coat protein goes where? Anybody heard my last year video or watched it? And you want to tell me? No? Oh, that makes it even more fun. Okay. So, what do you say? You see these guys over here? These cop-chew guys over here? Well, we call those things cop-chew, so now you can imagine two cops, right? Two cops are gonna have fun, so they go to the... Are there any cops in the class? No? No, right? Should I stop the recording? I don't want to get in trouble. Okay, they go to the Goji. Goji. Do you know what I mean? No, everybody PG-13 in this class. Orgy. Do you know what an orgy is? Exactly. So, why two cops are going to the Goji? Goji is the L, okay? It's my accent. I didn't say it, right? Two cops that you gotta go to the Goji. Now, what happens if you have only one cop? He gets depressed and he goes to the ER. Cannot join the Goji. Okay? So two cops go to have fun at the Goji. One cop gets depressed, goes back to the ER. What is the ER? Emergency room. Okay, you got the picture. It gets better. Okay. So, there goes Goji ER, blah, 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 blah. So here we go more with this story, okay? So two cops go to Goji, one cop goes back to the ER, and then cluttering is only involved in this movement over here. So you're gonna have Goji, and this is a trans Goji, right? Goji to endosome, or it's going to be involved in here, mainly import or endocytosis of things that were outside of the cell. So that's the main job for cluttering. So now we're going to see what happens. Okay. So basically, you have many types of protein codes. So two major classes. One of them is cluttering coated. The other class is contamer coated. That's where you have your COP1 and your COP2. So COP1 and 2, those are contamer type of coating proteins. All right. So let's see here what happens with the cluttering business first. Okay. So first you have this protein that is shaped like a boomerang. Okay. And this type of shape will allow self-assembly of the subunits. So when you look at the cluttering coated vesicle, you see this type of formation that kind of looks like a protein cage, or it looks like a virus capsule. Okay? Because if you took virology, you also know that viruses have proteins that do self-assembly, right? They can do the self-assembly all on their own, and they form that coat that protects the virus genetic material, right? So here's the same business with this cluttering. It can do self-assembly, but it does not work by itself. It needs adapter proteins, and it needs to feel that it's going to carry something. Okay? Because if the cluttering is just going to do the self-assembly without anything to carry, that's wasteful. Okay? So it has to get signals from cargo. They're going to interact with receptors. It has to use adaptive proteins, and then this other one that you're going to see in a minute what that is used for information of that. So here is your cluttering story. Okay, so let's see what is happening over here. In this part here, you're going to have this cytosol. So this is inside of the cell. Okay? And this part here is outside. Okay? So let's see what happens over here. You have a receptor that is pointing to the outside of the cell. Okay. If this receptor is empty, it doesn't need to work. Now let's say that the receptor found a molecule that is interesting for this cell, and the receptor wants to eat up that molecule. So the receptor is going to bind to the molecule. If it is only one receptor, it's useless, so most likely you don't have a lot of those molecules around to be eaten in the first place. But when you have several receptors that are interacting with these cargo molecules, then the receptors are going to start to do something. First thing it's going to do, it is going to modify the inside domain, because this is a protein, is going to modify after it binds this cargo, and the modification is going to allow interaction with this light green protein over there. This light green protein is what you call adaptin. So that is your adapter molecule. Once your receptor interacts with adaptin, adaptin is going to fall on clathrin subunits. That's your boomerang there. So adaptin interacts with clathrin, and then here you have more receptor interacting with adaptin, interacting with clathrin, and that is going to start the self-assembly thing of that clathrin molecule. Okay? And here, keep in mind, this is 2D, but this is really happening like a soap bubble that is getting formed. So this is really a 3D sphere that is starting to get shaped. Okay? So now what happens? You have more clathrin that assembles all around this piece of membrane. And then at the end, you're going to have only a little neck of this bubble that is connected to your plasma membrane. And clathrin somehow is not good at getting the job of separating this bubble from the rest of the membrane. So this is when you need a protein that is going to blow up that neck. So you call that protein dynamin. It makes sense. Hopefully, right? So dynamin gets there and squeezes that neck and separates this vesicle that is coated with clathrin. It separates the vesicle from the plasma membrane. So now endocytosis is taking place. Now, once this thing gets endocytosed, it should go to the endosome and then to the lysosome, right? But the problem here is that if this vesicle is coated with clathrin, then you are masking the receptor. That means this thing doesn't know where to go. So in order for the receptor to be showing over here, you have to dissociate the clathrin from this vesicle. So this is when you get this dissociation and you end up with a naked vesicle. Not totally naked, because it has those receptors that are going to direct it towards where it has to go. Okay? So everybody got the clathrin story? All right. Let's see here at other examples. Right? So here is a type of example in which you are going to have regulated secretion of something. Okay, regulated secretion. In what kind of situation could you have regulated secretion of something that we already have looked at in the class? Huh? Neurotransmitters. Which neurotransmitter? Glutamate, acetylcholine. Those are good, right? I was thinking acetylcholine, glutamate will do it. Neurotransmitter. Because neurotransmitters are packed in little vesicles, and then you can get a signal to get this vesicle to fuse to the membrane and dump this thing out in the synapse, right? Or to the outside, okay? So what happens here? You have adapter proteins that will receive a signal for controlled exocytosis. So in this case, you are having controlled exocytosis, not constitutive. Okay, so this is controlled. You need a signal in order for this thing to happen. Okay, so let's see what is important to know over here. So you have here one kind of Rab GTPase protein that when it is bound to GTP, it will go and associate with a specific part of that vesicle. Okay. Now, Rab with GTP can also associate with a molecule that is on the plasma membrane that is specific for interaction with that particular Rab. So when you say Rab, there are many, many Rabs out there. Okay? So this is going to be specific interaction because you want to have this specific cargo to be exocytosed on demand. So you're going to activate your specific Rab that is going to go there and allow vesicle fusion. Okay? So here is the mechanism of how that happens. Right? You have Rab with GTP that interacts with this tethering protein, and this tethering protein is going to bring this vesicle very, very close to the membrane, but it cannot make the vesicle fuse directly to the membrane. It's going to need other stuff. These other stuff are the snares. Snares. You're going to have V snares for the vesicle. You're going to have T snares for target membrane. Okay. So this adapter protein over here, this tethering protein, is going to bring the vesicle so close to the membrane that those snares are going to be able to detect each other, and they're going to make a little spaghetti shape interaction. They're going to intertwine. Okay? So once those snares are interacting, this interaction will bring this vesicle so close to the membrane that is going to allow fusion of that vesicle and exocytosis of your cargo. Okay? So things that are important over here. You have the Rab with GTP. You have tethering protein, and you have the snares. Okay? So here is showing how the snares interact, V and T snares. They're going to curl up here, and they're going to allow vesicle fusion. Okay? Examples of that. All right. So here is just another, just writing out what I have just mentioned. Okay. And here is more to the story. How is it the Rab is working? Right. So let's see what happens here. The different Rabs, those are the ones that are going to give specificity to those vesicles. Okay. So you're going to have very, very specific form of targeting. So the Rabs, they are GTPases, that means they bind to GTP, and once they are bound to GTP, they can hydrolyze GTP. If you hydrolyze GTP, you end up with GDP, right? So let's see what happens here. The Rabs are only active when they are bound to GTP. When is it, this part is the part that everyone misses. When is it that your Rab is going to get GTP? Well, only when you want them to be active. That's very redundant. Right? Look at this. The donor compartment has this protein over here that is specific for that Rab, and this guy happens to be a GAF. Remember what the GAFs do? G protein exchange factor. Where have you seen that before? Nuclear transport, right? So here is another GAF. This one is on the membrane, and is specific for the Rab. And this guy here is going to be activated, or is going to be placed here on the membrane strategically when you need this exocytosis to take place. So once your GAF is on the membrane, it is going to wait for soluble Rab that is floating around with GDP bound. So this is diffusing, gas over there interacts, and GAF is going to kick out the GTP, and is going to kick out the GDP, and is going to put in a GTP. Once you have GTP, this is when it's going to interact with your vesicle that is getting formed over there. Okay? So a whole lot of things is happening over here. So what happens here? Once you have your Rab GTP, this is when you're going to work with your adapter protein, which is right there. This is your tethering protein. It's also called the Rab effector. So two names for tethering protein. Okay, so just in case, make sure you put observation here, saying this guy here is also I can never spell it, protein. Okay, so here, what is happening? You're going to have your active form of Rab that was activated from the soluble form. Okay, then you're going to find your Rab effector, and you're going to bring this membrane, this vesicle close to the membrane, and get your snare business going on over there. Okay? All right. So I mentioned we have different types of Rabs. Okay. So for example, you have Rab1 that is important for ER2 Golgi. Okay, so this is adapter molecule. You have Rab5 that is important for early endosome formation, but depends, we have all different types of Rabs. So this is just to tell you, you have specificity. If you want to find more about the video where I got these pictures, you can follow that. All right, so let's see what is happening here. After you have your vesicle fusion and you got your exocytosis, in this case here you have a presynaptic neuron and here's your postsynaptic neuron, right? So you have exocytosis. Now you end up with the snares that are all tangled up on the sides. They do not untangle by themselves. Okay, guys, let's be quiet, please, right? So they do not do the untangling by themselves. They need help. So specifically, they need two proteins. They need this guy that is called NSF, and the other guy is alpha SNAP. It's easy to associate SNAP to snapping the spaghetti away from each other. Okay, so those guys, they have to untangle these snares. So they are going to use energy to break those things apart. They are going to use ATP energy. Okay, after using ATP energy, those spaghetti balls here are going to be pried apart from each other, and they're going to get recycled, right? So to bring those snares away from each other, you do need to use ATP energy. And so here is the next part. Let's see where we're going here. All right, so here, more details on your COP2 type of action. So first thing you have to do vesicle budding, right? And this is from the ER. You have to do the budding, and then you do the uncoating and fusion with your target at the cis Golgi. Okay? How is it that you do the budding? This is happening in a similar way as we have seen with clathrin, but with details on the different proteins that get involved. Okay. First thing you're going to have over here is a GTP binding protein. Okay. And also you're going to have those adapter molecules, those cotmers. Okay. So once you form those adapter molecules, and this is in analogy to your clathrin molecule. Okay. So after that, you are going to allow fusion. Okay, still going to take away the coat, and you're going to allow fusion and delivery of your cargo, and you're also going to use the snare mechanism to fuse the membranes. Okay, so that's one thing that has in common. So what are those coat proteins? What are the adapter proteins? Because COP2 is a type of vesicle, but it's not telling you the name of the proteins that are involved in this vesicle. Okay, COP1, same thing, type of vesicle. So here, for the COP2 vesicles, you have certain coat proteins, and those are the ones that are easy to remember because you look at the name of each one of them. Anybody want to tell me what are those names? Hello, what are the names of the adapter proteins? sec 23, sec 24, sec 13, sec 31, sec 16. I don't know what sex they are, but they are having a lot of sex in there. I tell you, those cockatoos are always fun. Okay? Oh, I got somebody who had a light bulb. It's not S-E-X, it is S-E-C. Okay, it's not spelled wrongly. Okay, sec, not sex. Okay, so cockatoo have a lot of sex, and and on top of everything, they got these associated GTPase. It's called SAR. I don't know what the SAR is for. All right. But SAR, it sounds like almost like sex. So SAR, sex, and and everybody goes to Golgi. Okay. So we get more story about these guys. Now let's look at the boring guys, the COP1 guys. They have all different forms of coattamer proteins, right? I don't remember if I put some specific ones here, but they are definitely not having any of this sex. That's why they get depressed. Okay. Instead, they are having also. They also have to have this GTPase adapter protein, and they are so bored, and they go arf. That's the name of the associated GTPase for these boring guys. Okay, arf. All right. So that is your retrograde transport. Okay. Now let's see what these proteins, how they are doing all this interaction. Okay. So here, of course, we're going to start with the COP2, right? So what is happening over here? First thing that you're going to find, this is on your ER membrane, right? First thing that you're going to find is that GTP exchange factor. Remember that GTP exchange factor? This guy is going to go to the membrane once you want to make vesicles and transport stuff to Golgi. Okay? So your GAF gets in there. The name of your GAF is SAR. Okay, SAR1 GAF. It gets in there. Once it is on the membrane, it is going to do the gaffing thing. Okay, that means it's going to get GDP kicked out of adaptive GTPase, and it's going to put GTP on it. Okay, so let's see more of that story. So you got your SAR, which is a GAF, right? And you're going to have a receptor for that. Okay. Then the receptor here is going to be one of those adaptive proteins. In this case, it's a sex something. All right. So sex something gets there to your SAR, and the sex is going to get a powerful GTP. It's going to get energy, right? What is going to happen here? Your SAR is going to show a very interesting hydrophobic tail, okay, on your sex protein, right? So SAR is over here, it gets the GTP, is going to get here interaction with this sex protein, is going to expose a hydrophobic tail that is going to get inserted in the membrane. Okay. Now, what is going to happen over here? Okay, here, sorry, here is your sex protein here. The tail gets inserted into the membrane, and something is supposed to happen over here. Okay. This SAR1 starts to curve the membrane, and curving the membrane is going to allow more interactions of SAR, okay, with the adapter protein that is going to start to make up the bubble, just like you have seen with the clattering. Okay, so let me see if I have a better picture of that. Okay. All right, so let's see here if this clarifies a bit more. So SAR1 is going to interact with coat proteins. SAR1 is over here. is going to interact with adapter proteins. SAR1 is going to stick a piece of the protein in the membrane, is going to allow curvature, and then you have assembly coat proteins that are going to make this thing to bubble out of the membrane. To get away from the membrane, this thing is not showing you here the dynamine. So yeah, I'm not sure about the protein that does that separation. But I think this is enough detail for that. So what happens here? You have still this protein here that was activated by GTP and is allowing the interaction of those sec coating proteins, right? But this guy here is a GTPase, so it's going to hydrolyze the GTP. Once the GTP is hydrolyzed, the coating proteins are going to be dissociated. Okay. So this one gets dissociated, and here you're going to get your naked vesicle that is going to go towards the Golgi. Okay? So it's a basic type of mechanism, and what you need to keep in mind is when is it that you have GTP on SAR, when is it that you get association with the membrane, and when you get vesicle formation based on those adaptive proteins. So here is another picture straight from your book showing how that is happening. Okay. So here you have your SAR1 with GTP that is bound. You have your exchange factor that put GTP on SAR1. And you get your SAR1 interacting with your coat proteins, with a lot of, the sec proteins. Oh, finally, you got a question. All right, get your points here. I wonder what goes on the confidence level of this one. What is SAR1? See, everybody's sharp today. 25 seconds. Does anyone need more time? No? Okay, what is SAR1? It is a G protein. It is a protein that associates with GTP. All right. So here is just another picture showing in more details your associated coat proteins. Okay. So remember, here you have your SAR1 exchange factor, that is a receptor, and is going to put GTP on SAR1. SAR1 inserts itself on the membrane and it starts to gather the associated protein that makes the coat. And then the thing buds out. Okay. Oh, another question. COP2, coated vesicles, move the material from where to where else? That's the one question that everybody gets right. Still, I have some people giving me the wrong answer. What is it you have to remember here? 2 cups. Anybody need more time? Okay, I'm stopping it. What is your answer? C. 2 cups always go to the Golgi. All right. So now we're going to look a little bit at your part 1. because they cannot be ignored. So similar story, different adapter proteins. All right. So here you have your G protein exchange factor that is nothing to do with SARS or sex or anything. This one is the ARF guy. All right. So your ARF exchange factor is going to put GTP on ARF. ARF is a GTPase that is floating around when it's bound to GDP. When it gets GTP, it is going to interact with the membrane. So ARF gets activated by the ARF-GAF, gets into the membrane, and it does the same business as the SAR. It's going to get the other stuff together over here, okay, the other adapter proteins, and it's going to start making the bubble. And then you're going to get the bubble out of there, you get your vesicle formation. And you get another question, just to compensate. The last time I didn't give you a lot of questions. Cup 1, coated vesicles. Move the material in what direction? I had intended to put a radical direction for C, but oh well. Okay, 5 seconds. Does anyone need more time? Remember, anterograde is forward, retrograde is backwards. What is your answer? A. That is your retrograde. Okay. More? Oh man, I'm really generous today. I didn't remember this guy. All right. RF1 is what? All right. Who needs more time? By answer? Okay, what is R1? It's a GTP binding protein. Okay. Now this question is a little bit tricky, but you know I don't like tricky questions, so I'll not put this on an exam, right? What else can be associated with RF1? GDP. But it's not binding to it, it just hydrolyzes GTP. But that's very tricky. I would have a headache if I would ask such a question. Okay? But you're right. GTP is the binding protein. All right, so when you're making proteins that are going to be exocytosed, usually those proteins are going to undergo some type of modification, right? So that happens usually in the Golgi. What kind of modifications? One type is glycosylation. So this type is adding sugars or carbohydrates to your protein, right? So you get modifications. These proteins will get exocytosed, okay? And there are, okay, this is a part that is also very, very important to remember, okay? So this is a part that also we're going to correlate with some diseases. So you have two types of vesicular secretory pathways. One way is the way that we are talking about, that is a regulated type of secretion. It is on demand, okay? There are other types of secretion that they're actually happening all the time. So those are constitutive types of secretion. So vesicles that are usually being exocytosed, so that is constitutive, right? Is in comparison to regulated secretion. Now, look at this. Look what it says over here. When you have proteins that do not have specific receptors that are destined to go somewhere, those proteins are always put in the default mode of constitutive secretion. Okay? So if a protein is made in the ER, it goes through Golgi, and it doesn't have a specific destination, then it's going to be dumped outside of the cell. And that's a pretty good pathway, is very useful, because let's say, for example, something wrong happened, you have a genetic mutation, your protein is not being made properly, and you didn't get the correct zip code for that protein, you didn't get the correct destination for the protein, correct vesicle formation, then what's going to happen to the protein? If the protein does not undergo the default pathway, then this thing is going to be accumulating inside of the cell, and that's bad news. Okay? That could really cause cell damage. So whenever you have a protein that is not processed properly, doesn't have a destination, then it is always going to go through the default secretory pathway. Okay. All right, so here is a summary of those things I've just talked about. So here a little bit on endocytic pathway. Okay, so this is the guy that is going to use clathrin. So here is your clathrin coat. Okay, same way that you used from trans Golgi, that's going to go to your lysosome. This thing is coming from the cytoplasm. Okay, that we looked before over here, and is going to be brought to the endosome. Endosome is going to fuse with the lysosome, and this thing is going to get digested. Okay, guys, let's be quiet, let's pay attention, because something interesting is going to come up over here. Okay, so once you have fusion of endosome with lysosome, this thing is going to be digested, and you can use whatever the thing ate, or you can recycle products. Okay, so lysozymes are the enzymes present in the lysosome. Now let's look here. All right, so this is also what I had mentioned before about constitutive pathway and regulated pathway. So this is just to have something on the paper, that you can study from. Okay. Let me see what else is important here. If I can go straight to the disease site. Okay, we had mentioned before. Okay. All right, one thing that I did not mention before, when you look in the Golgi, you see that you have these cis Golgi vesicles that fuse to the real Golgi, and the Golgi has a lot of these sacs structure. Each one of these sacs is called a cisterna. Okay, so cisterna. That's where proteins get modified. All right, so this is the path for constitutive secretion. Okay, this is a cool thing. Look at this. You have your proteins that get from Golgi from ER to Golgi, and then in the Golgi you're going to have the formation of vesicles that should also interact here with motor proteins on microtubules, and then those little vesicles are going to be brought to the membrane, to the cell membrane, and they're going to get exocytosis. Okay, so let me see if I have anything else written in here. Okay, this is the important part. It is going to travel using these motor molecules. When the motor molecule is going towards the cell membrane, those motor molecules are called kinases. When something is traveling the opposite way, those are dynamines, I think. Okay, now I forgot if I did not write that down. Kinesins are the ones that are going towards the membrane. So let's see here. All right, so this is the cool part. In the lysosomes, what is going to happen in the lysosomes? You're going to have digestion. You're going to have a pH that is very much acidic, okay? And you are going to maintain the pH gradient by usage of energy. Remember the guy we mentioned before? hydrogen ATPases, proton pumps. Okay, so proton pumps are going to be in those lysosomes. They are going to keep on packing hydrogens in the lysosome to maintain that very, very acidic pH. Now, what are the proteins that are destined to go to such an acidic environment? So those are the ones that are going to be delivered from the trans-Golgi to those lysosomes. Okay? And the coating part is going to be by clathrin. Okay, clathrin coat. Okay, so here is a part that I want to mention to you. Okay. So lysosomes, they are very much important to degrade things that are not useful to the cell anymore and to recycle material. But sometimes you have proteins that are digestive proteins, that are enzymes, that they do not work properly and they do not do the job that they're supposed to do. One enzyme that has that job is called hexaprotein, hexaminidase. I have to check on this spelling. Does that remind you of anything? No. Remember those gangliosides? Yeah. I think it is hexaminidase. This is an enzyme that degrades gangliosides. Remember the gangliosides, what they are? Lipids. They have a lot of sugars. Right? Remember those lipids? No. Go back and look at those lipids. So gangliosides, they have fatty acids, they have a certain structure, they have a lot of sugars that are really making up a lot of stuff over here. Okay? Now, I got 3 more minutes here. When hexaminidase does not work properly, when it works properly, you break down this guy, you get little pieces, and you recycle stuff. When hexaminidase does not work properly, what's going to happen? This sounds like a test question. Yeah, that's Tay-Sachs disease. But what is the molecular mechanism of Tay-Sachs disease? The molecule over here does not work. The protein does not work. You cannot break down those lipids. They get accumulated in the lysosome. And this is when your lysosomes get bigger and bigger and bigger and ends up destroying the cell. If the cell is a neuron, that's brain damage. Okay? So this is a type of a disease that you call lysosomal storage disease. Okay? Everybody got that? Lysosomal storage disease or diseases. Okay? So the entire process, meaning that you have to go through this vesicular transport, you have to dump it in the lysosome, and the molecule has to work. You see that it's getting together with other things that we have seen before, like with degradation of lipids or other stuff, for recycling and proper cell function. Another very important job of lysosomes is to recycle mitochondria. That is what you call mitophagy, or general term is autophagy. When you recycle damaged mitochondria and you are able to clean up the cell. Okay, everybody cool? Yeah, do we have questions? Yeah. Treatable, which part? Most of them no. No, this is different. Although it is involved, lysosomal processes are involved in aging, but lysosomal storage diseases are not quite involved in aging, as far as I can remember. It is something else. Yeah. Oh yeah, Tay Sachs is little. It will kill before 3 years old, sometimes 5. Yeah, it's very little.